Good morning, students. Welcome back to another Waidsum Academy video. Today we're doing IGCSE Biology, Chapter 18, Inheritance. In this chapter, we're going to learn about DNA, what are chromosomes, genes, proteins, and we're also going to learn about mitosis and meiosis, and we're also going to understand the difference between genotype and phenotype. So let's begin. In section 18.1 DNA, we're going to learn about DNA and what's the sequence of bases and explain the different sequence of amino acids and protein and so on. So, how is DNA organized inside the cell? DNA, or deoxyribonucleic acid, is a molecule that carries genetic information. The information carried by DNA is important for all cellular functions, such as cell division and cell differentiation. Thus, almost all cells in our body contain DNA inside their nuclei, and about 2 meters of DNA can be found in each cell nucleus. Remember, in our previous chapter, chapter 17, we learned about the female gametes and the male gametes must combine to form the zygote. Well, this is actually a process of meiosis. Well, we're going to learn about meiosis in this chapter. How does meiosis actually happen? Well, it actually happens because of DNA. DNA is what allows it carries the information that is important for cellular functions. So now, how are DNA organized in the cell? Now, if you look at this image, you see there's this DNA, this image, you could see that there are 2 strands with a bunch of color combining the strand, and as they go on and to be magnified. Well, let me explain. A small segment of DNA carries a gene that stores information used to make a single protein. Proteins are responsible for determining the characteristics of an organ. So, when you see somebody, they have, or let's say a person has black hair, light skin, blue eyes, freckles, long fingers, etc. These are all proteins determining the characteristics of our organs. The gene is what stores the information used to make a single protein, and the proteins, they describe an organism, they explain the characteristics. Now, each DNA molecule consists of 2 strands twisted around each other to form a double helix. You see these two strands. These 2 long strands right here, these 2 strands combining to form this spiral shape. Well, the 2 of them are what forms the double helix. They twist around each other, and each segment is a gene. As you can see, there's gene 1 and gene 2. Now, as it goes on to become magnified, you will see that a molecule of DNA is wrapped around each protein to form a single chromatic thread. Now, you see as they magnify in the helix, you see that it's wrapped around the proteins. Now, what's wrapped around the proteins? The chromatic thread. The chromatic thread is what is wrapped around each protein. This chromatic thread, this thread in the DNA double helix, is wrapped around protein. And during cell division, the chromatic threads coil tightly into a thick rod shape. That means during the cell division, the chromatic threads that surround the protein, the chromatic threads that surround the protein are coiled tightly into a thick rod-shaped structure inside the nucleus. These structures are called chromosomes. So inside the nucleus, the chromatic threads, they tightly coil around each other to form the chromosomes. These structures are called the chromosomes, and these chromosomes are found inside the cell, inside the nucleus of a cell. So let's move on to how are chromosomes, genes, and proteins related. Now that we understand what a chromosome, gene, and protein are, we need to understand how they're related. We have seen that chromosomes consist of DNA molecules coiled around proteins. The DNA molecule consists of two parallel strands, as you can see, and they coil around the proteins. The two strands are joined together by a nitrogen containing on a nitrogenous basis. There are four bases: adenine, cytosine, guanine, and thymine, which is A, C, G, and T. And they each have their complementary bases. Adenine always combines with thymine, and cytosine always combines with guanine. You can never have an adenine and a cytosine combined together. These two cannot bear each other. These two cannot combine with each other. It's only with adenine and thymine and cytosine with guanine. This is the only way these are the only two complementary bases. So in the IGCSE questions, if they ask you, is it possible for cytosine to combine with adenine, you say no, it is not possible, because they cannot be complementary bases. Now these four bases are what join the two parallel strands together. These strands of the DNA, this is what these bases are combining together, connecting together. An uncoiled DNA molecule can be represented by a simple model shown in this diagram. The molecule resembles a ladder, and the twist of the coiled DNA molecule represents a spiral staircase or a double helix. Now, the two strands that are joined together, such that A is joined to T and T is joined to T. As you can see, A and T are always combined together. A and T, since A is the green, and in this case A is green and T is red, A and T are combined together. While C is blue and G is yellow, C and G are combined together. Now, it's because of this, they each have their complementary shape that helps them lock together, click together, and their shapes are complementary to each other. That's why you cannot combine A to C or G to T or whatever. So now let's move on to understand genes. Our chromosomes, as we still explain how our chromosomes and genes properly related, we'll explain gene. A DNA molecule may carry genes along its length. Each gene is a segment in the DNA that controls the production of a particular protein or an enzyme, membrane carriers, or neurotransmitters. So each DNA molecule carries a gene along its length, and the gene is a segment that controls the production. So it controls the amount of proteins, enzymes, membrane carriers, or neurotransmitters produced. And the DNA molecules carry many of these genes along the length. Remember when I explained that each segment of the DNA is a gene? This is what I mean when I say so. The genes are carried along the DNA length, and they each controls the production of proteins, enzyme membranes, and neurotransmitters. This is what you should memorize: proteins, enzymes, membrane carriers, or neurotransmitters. So let's move on to protein synthesis. Now, proteins are made through a two-step process: synthesis of mRNA synthesis of M R N A, and the synthesis of a protein. They are made through the step process. Now, what are the synthesis of an mRNA? Well, we'll learn in this diagram. This diagram shows. Now, you see the DNA template. There are two DNA strands unzipped. There's two DNA strands unzipped, and the synthesis of mRNA in the nucleus. The region of DNA carrying two genes unwinds and unzips. The two DNA strands are separated, as shown in the diagram. Now, one of the DNA strands, called the template, is used to make the messenger and RNA, which is mRNA. The M in mRNA stands for messenger. So, one of the DNA strands, called the template, is used to make the messenger RNA, which is mRNA. The mRNA copies the message in the gene. The mRNA carrying a copy of the gene leaves the nucleus through a nuclear pore. So inside the cell, we have this nucleus, the cytoplasm, and the nuclear pore. Now the mRNA leaves the nucleus through the nuclear pore, and it arrives here. That's the step 2, the synthesis of protein in the cytoplasm. In the cytoplasm, the mRNA attaches to the ribosome. The ribosome translates the message in the mRNA into a sequence of amino acids. Amino acids are proteins, are smaller units of proteins. Now, the ribosome translates this mRNA message into amino acids. These amino acids are joined together to form a protein. As the ribosome moves along the mRNA, more amino acids are joined together and the protein made gets longer. At the end of the mRNA strand, the ribosome detaches from the mRNA and the protein is released. The order of or sequence of amino acid in a protein molecule depends on the sequence of bases of the mRNA. The sequence of bases on the mRNA in turn depends on the sequence of bases in the DNA. So the mRNA, it has two parts. The protein synthesis has two parts. The synthesis of mRNA in the nucleus and the synthesis of proteins in the cytoplasm. The mRNA has a message as the DNA unstrands and unzips, and it takes the message, and then mRNA leaves them with the content holding the message, leaves the nucleus through the nuclear pore and arrives at the ribosome at the cytoplasm. The cytoplasm translates these messages to a code of amino acids, which creates a protein. The the ribosome translates the mRNA to a coding which contains the protein, and it's the protein that explains the characteristics of a organism. Proteins are responsible for determining the characteristics of an organism. So let's move on. How do genes control cell functions? The nucleus in every cell of the body contains all the genes necessary to construct the human body. So if so, how can there be different cells and tissues in the body? Well, genes can be switched on and off, and if only the muscle genes are switched on, then the cell will become a muscle cell. Similarly, if the nerve cell genes are switched on, a nerve cell will be produced, and this is a process of differentiation. So this is something you should also memorize for your IGCSE. They're explaining that since there are different types of cells in the human body, a gene can be switched on or off. And if an only muscle gene was to switch on in a cell, the cell will become a muscle cell. And it will be same for the nerve cell genes. If they're switched on, a nerve cell will produce, and so on. So here are some questions for section 18.1. You can pause this video and try to solve them, and try understand them if you could. So we could move we can move on. These are typical questions you will see in the IGCSE. So let's move on so we could cover up this chapter. Now we're in 18.2, mitosis. In this section, we'll learn about mitosis and the role of mitosis in asexual reproduction. So let's move on. What is mitosis? A mitosis is a type of a nuclear division. It produces a daughter nuclei which contains the same number of chromosomes as the parent nucleus. For multicellular organisms to grow, its cell must divide. The type of cell division responsible for growth and repair of worn-out tissues is called mitosis. The nucleus in each cell contains chromosomes, and each chromosome is made up of a DNA molecule which carries many genes along its length. Each gene controls one particular characteristic in our body. For example, one gene gives us a particular hair color, and another gene controls the way we roll our tongue. So mitosis is the nuclear division that produces a daughter nuclei which contains the same number of chromosomes as the parent nuclei. As you can see in the diagram, here are nucleus containing two chromosomes. Each chromosome divides into two daughter chromosomes attached together. The chromosomes line up along the equator of the cell, and the daughter chromosomes separate and move apart. Two identical daughter nuclei are formed, and the cell divides in each containing two chromosomes. So you can see, despite the fact that the nucleus divided into two, the amount of chromosomes each cell has is the same as the parent cell. How this happens is because the chromosomes line along the equator of the cell, and the daughter cell chromosomes separate and move apart. That means they split into half, containing two. So two identical daughter nuclei are formed, and as the cell divides, each contains its own two chromosomes. So this entire process is known as mitosis. Now we'll move on to what are stem cells and the importance of mitosis. Now every organ has a certain unspecialized cell called stem cells. They are able to divide or produce new cells by mitosis. The daughter cells produced can change to form specialized cells for specific functions in the organ where they are found. For example, if a person has donated a part of his liver, the stem cells then divide to regenerate that part of the liver that was removed. So basically what this means is, let's imagine there's an organ like a liver. To understand this, you need to understand that each organ has its own type of cells. There's blood cells, there's liver cells, there's the white blood cells, and so on and so forth. There are many cells in the body that each help with building the organs. Now, what are stem cells then? Stem cells are basically cells that can help replace their own specialized. That means if a cell were to get damaged, stem cells could replace that cell and pretend to be its role, act as its role. So if a person were to donate part of his liver or her liver, if a person were to do so, a stem cell could help regenerate that part that has been donated. That's why, because they are unspecialized. They don't have its main function. They are cells used to modify and also replace cells like skin cell, liver cell, blood cell, and so on. And they are also capable of mitosis, although stem cells are collected in the embryo. Now, another example is the production of red blood cells in the red bone marrow. To produce a red blood cell, these changes occur. 1. The hemoglobin is produced in the stem cell. 2. Cells divide to produce two cells of different sizes. 3. The smaller cell that contains the nucleus is broken down. And 4. The larger cell has no nucleus. The mitochondria inside this cell are broken down. the cell becomes a red blood cell with a biocave shape. So what is the importance of mitosis? Why is mitosis important in our body? Mitosis actually ensures that the resulting two cells are genetically identical to the parent cell. This is important for processes such as the growth in multicellular organisms, repair of damaged tissues, and asexual reproduction. We learned in chapter 16 of the reproduction in plants what asexual reproduction is. Asexual reproduction is a reproduction that only contains of one parent. So mitosis is important in this process, in the same also as repair of damaged tissues and growth in multicellular organisms. And stem cells too, because stem cells can also divide and produce new cells by mitosis. So let's move on to section 18.3, which is meiosis. What is meiosis and what are we going to learn in this section? We are going to learn the meiosis as a cell division and describe the haploid or diploid nucleus. So let's move on. What is meiosis? Well, sexual reproduction in organisms involves the fusion of two sex cells called gametes. In humans, the male gamete is the sperm and the female gamete is the egg. Now, gametes contain half the number of chromosomes as the normal body cells. Gametes are genetically different. This is because a reproduction division called meiosis occurs during the gamete formation. Meiosis is a type of nuclear division, and the parent nucleus divides to form two daughter nuclei. Each daughter nucleus contains half the number of chromosomes as the parent nucleus. So remember during mitosis, in mitosis we said that the daughter nucleus has the same number of chromosomes as the parent nucleus. But this is not the same as meiosis. In the meiosis, each daughter nucleus contains the half the number, not the same half. It contains half of the number of chromosomes as the parent nucleus. Consider an animal cell containing two pairs of chromosomes, as shown in the diagram. The chromosomes are shown in blue inherited by its father, while the chromosomes shown in red is inherited by its mother. As you can see, there's two chromosomes. Here says the blue eye gene and the black hair gene, and the brown eye gene and the red hair gene. Now the blue shown is inherited by the father, and the red chromosomes, are inherited by the mother. So now there are possible different combinations of parental chromosomes. That means there are different possibilities of a child collecting some of the father's chromosomes or some of its mother's chromosomes. Example, there's a 25% chance that the child born will have blue eyes and black hair. There's a 25% chance that the child will have blue eyes and red hair. There's another 25% chance that the child will have brown eyes and red hair. Brown eyes and black hair, I mean. And there's a 25% chance that you have brown eyes and red hair. Now, when a child, these all codings are different gametes, 4 genetically different gametes. And meiosis is this type of cell division that creates these gametes. That means it produces daughter nuclei which contains half of the number of chromosomes as the parent nucleus. Notice as the parent nucleus both have 4, but each child has only 2. This is the process of meiosis. Meiosis produces 4 genetically different gametes. So let's move on to the difference between haploid nucleus and a diploid nucleus. Let's consider a sperm or an egg. Each contains one set of 3 chromosomes, as shown in the diagram. During fertilization, a sperm fuses with an egg. The fertilized egg or a zygote produced will contain two sets of chromosomes. Hence, the zygote has three pairs of chromosomes. One set of an unpaired chromosome is called a haploid number. That means one set of an unpaired chromosome is called haploid. So a sperm cell on its own is a haploid cell, and an egg cell on its own is also considered a haploid cell. Now, the zygote that is formed by the fusion of the egg and sperm cell is known as a diploid cell. Why? Because it contains the number of paired chromosomes. That means both the chromosomes of the egg cell and the sperm cell, when they fuse together, they form a diploid cell. Now, this diploid cell is this zygote. This diploid cell is known as a zygote. Now you want the difference between a haploid nucleus and a diploid nucleus. A haploid nucleus only has a set of unpaired chromosomes, while the diploid nucleus has the number of paired nucleus. As you can see in the name, di means two. So just say a haploid nucleus has one set of unpaired chromosomes, while a diploid has a set of paired nucleus. Now, what is the importance of meiosis? The importance of meiosis. Meiosis produces haplogametes. During fertilization, the nucleus of the male gamete fuses with the nucleus of the female gamete. The normal diploid number of the chromosomes is restored in the zygote. Meiosis produces gametes that are genetically different. So this is something you should understand, that during fertilization, when the nucleus of the male and the female gamete fuses, the normal diploid number of the chromosome is restored in the zygote. That means the meiosis produces gametes that are genetically different. Random fertilization produces even more variations in the offspring. The greater the genetic variation, the better the species is adapted to changes in the environment. For example, if the organism of a species is genetically similar, any change in the environment may wipe out the organisms of a species of a whole population. That is why meiosis is not found in asexual reproduction, because in asexual reproduction, they are all genetically the same. So if a disease were to form or a change in the environment were created, the entire population got wiped out. But it's not the same in meiosis. In meiosis, they're genetically different. As explained, they're genetically different, so they can survive any change in the environment. But if they are genetically different, they may wipe out a whole population. If one organism dies because of the change of the environment, all the organisms within the same species will also die. If the organisms of the same species are so varied, it is unlikely that a change in the environment will destroy the whole species. Organisms with the genes that can give them a selective advantage in a new environment will survive. Those that survive will pass out on their favorable genes to their offspring. So, you should understand that the importance are meiosis produces haploid gametes for fertilization, and meiosis produces gametes that are genetically different. This is important as they might explain to you what does this mean. It means that they can produce organisms that are genetically different. So that can help them, they can produce genes that varies that helps them survive changes in the environment. It also gives them a selective advantages, and they can pass on these genes, these favorable genes, to their offspring. Now here are some questions. The figure shows a cell with pairs of two chromosomes. Four genetically different gametes are produced during meiosis. Draw in the chromosomes to show how the four different gametes are produced. Now you can pause this video and try these questions by yourself. You can try and see if you can get them, because these are common questions that will come out in the IGCSE exam. So let's move on to explain the monohybrid inheritance in section 18.4. So in this section, we'll describe this inheritance as a transmission of a genetic information from generation to generation. So how are traits passed on from one generation to another? As you can see in this picture, you can see some human traits can be inherited. In the father, he has hair type straight, wavy or curly, the earlobe attached or detached, or skin color fair, dark, etc. And these traits can be carried on by the child, as the kid can also have similar hair types, similar earlobes, or similar skin color, and they also have with their own traits, like eyelids if they're single or double, or the chin, which is with or without a cleft. The color of your eye, height, or skin color as traits are and characteristics that have been passed on from us from your parents. Our parents. They are referred to as hereditary traits because they can be passed on or inherited from one generation to another. However, for many centuries it was observed that traits could be inherited. However, scientists were unable to explain how such traits were inherited until the middle of 19th century. And these traits could be explained by genetics. What is genetics? Genetics is the scientific study of heredity, which is the traits. Heredity is the traits that you earn from your parents. Scientists who study heredity are called geneticists. Geneticists are interested in how traits are passed on from parents to their offspring. So what's a mono? So basically, geneticists are scientists who study genetics, and genetics is the scientific study of heredity. So what's the monohybrid inheritance? Monohybrid inheritance is the inheritance involving one pair of contrasting traits such as single or double eyelids, straight or curly hair, and attached of detached earlobes. In the 19th century, Gregor Mendel first explained how heredity might work. Mendel carried out breeding experiments on garden pea plants. His laws on genetics are true today, and now he is known as Gregor Mendel, the father of genetics. Just a little bit of history. Now we move on to genotype and phenotype. Now genotype and phenotype is the genotype and phenotype. We're going to understand what is genotype and phenotype, and allele, and dominant and recessive allele, and what are they basically, and how are they important in inheritance? What are alleles? Let's start with that. Our body consists of two sets of chromosomes, having inherited one set from each of our parents. Hence, each chromosome exists as a pair in a cell. The two chromosomes in each pair have the same shape and same genes and same sequence of the parent's genes. They are called homologous chromosomes. We obtain one homologous chromosomes from our father and the other from our mother. So then what are alleles? Each gene may exist in two different forms. The different forms of the gene are called alleles. For example, the eyelid gene has two different alleles. If we have a double eyelid or a single eyelid, one allele gives you double eyelid, whereas the other allele gives you single eyelid. If you inherited two different alleles from your parents, you will have double alleles. This is because when a person has two different alleles, only one allele will be expressed. In this case, the double allele is expressed, while the single allele is not expressed. And if a person has two different alleles, the allele that is expressed is called the dominant allele, while the allele that is not expressed is called the recessive allele. In this case, the dominant allele is the double eyelid, and the double eyelid is the dominant allele. Why is that? Because the double eyelid is expressed. That is why we say the double eyelid is dominant. Are recessive a recessive alleles ever expressed? So since we know that dominant alleles are always the alleles that express, are recessive alleles always expressed? Well, if a person has both recessive alleles, it will be expressed. Hence, if you inherited two recessive alleles of the same allele gene, then you will have single alleles. So let's move on to explain what are genotypes and phenotypes. What are genotypes and phenotypes? If a person has recessive genes, then recessive phenotype will be expressed. Different combinations of allele are called genotype. So genotype are the combinations of different alleles, and if they express characteristics of outward appearance are called phenotype. Example, capital letter D is known as the dominant allele, while the recessive is called the small letter d, the lowercase d. So now to help you understand, I'm going to write this down. And a person has a coding for when a person has a coding for a specific characteristic, we have genotypes that can explain which is expressed in the body. So example, let's say if a person has a chance of getting a double eyelid. In this case, the double eyelid is dominant, while the single eyelid is recessive. So type of which type of eyelid is expressed? Well, in DD, this DD means the double eyelid, and the double eyelid is the dominant eyelid. So which is expressed? Obviously the double eyelid, since both are dominant. And in the uppercase D and lowercase d, the double eyelid is still expressed. Why? Because the double eyelid is the dominant eyelid. It is the dominant eyelid. So in the eyelid, the dominant one is always the one that gets expressed. So it's not the single eyelid. It's only when both eyelids are recessive, then you can say the single eyelid is expressed. But if there's a dominant allele in the genotype, then the dominant is always expressed. Different people have different combinations of alleles called genotype. The expressed characteristics or outward appearance is called the phenotype, as shown in the table. Notice that the dominant alleles will always be expressed when it's present. That is true because the double capital letter upwards letter D, the uppercase letter D is always expressed because it's dominant. Now we're going to homozygous and heterozygous. A pure breed or homozygous organism has two identical alleles, DD or DD. Homozygous means same. So these two here are homozygous. Why? Because they're both the same. Uppercase D or lowercase d, both are DD, so it's homozygous. A homozygous dominant organism has two dominant alleles, while a homozygous recessive organism has two recessive alleles. Heterozygous organism has different alleles. That means this is hetero. This of the uppercase D and lowercase d is hetero. That means there's two different alleles. Now the heterozygous organism has two different alleles. Now the heterozygous is also the dominant because the dominant double eyelid dominant allele is present in the genotype. So that means it is dominant in the person who gets a double eyelid. But however, for the hormone which are same is different. One is dominant while the other is recessive. So let's move on now that we've understand this. Now let's go to genetic crossing. So let's use examples to show how characteristics are inherited. A purebred tall pea plant was crossed with a purebred dwarf pea plant. The first generation of offspring is called the first filial generation, which is F1 generation. When the F1 plants were crossed amongst themselves, the second filial generation, F2 generation, of the offspring produced were in ratio 3 tall plants, 1 dwarf plant. Explanation. So, see, here we did genetic crossing. They genetic crossed. Genetic crossing is a type of diagram we use to explain the chances of getting specific genes. Now you can see that the two pea pods were fertilized together, and the tall one pea pod is quite tall while the other is short. Now the tall is dominant, as you can see, because it has the dominant. Its uppercase letter is dominant. That means it is dominant in the genotype. This is a genotype. This entire thing is a genotype, and you can see that since it's uppercase, it is known to be genotype. And this is also homozygous since both are same. Now you can see that since both are same, this is also homozygous. Now in the dwarf is also homozygous, as you can see, but they're both recessive. Now, since both are recessive, when they combine together in the F1 generation, you can see that there's a chance the ratio for tall to short. There's a chance that all the child are tall in the F1 genotype, in the F1 generation, which is the filial generation. You know why all of them are tall? Because in each genotype, the dominant allele is present. That proves that the dominant allele is the tall one. And because of that, because they are all present in each genotype, all of the child are tall. All of the child are tall. Now in the second generation, they mixed in two child. They mixed in two different childs with the same. Now these two are heterozygous. All of the childs are heterozygous, and the next generation, both parents are heterozygous. So since the parents are heterozygous, there's a chance that you can produce a dwarf child. So in this case, the ratio is 3 tall, 1 dwarf. Now one has both dominant genes. One child has dominant genes, while one is a homozygous dominant gene, while two are heterozygous mid genes, because one is dominant, one is recessive, while the last one is homozygous recessive genes, and means it contains the same genes of recessive. Now that is it when we explain genetic crossing. It shows there's a chance for ratio type of the second. This is called a Punnett square, by the way, and the ratio type of the Punnett squares, the test results. To show the test result, the ratio type of phenotype is 3 tall, 1 dwarf. Now you can see that the tall has two T's, dominant, while there's the homo recessive the dwarf has homo recessive T's. So let's move on to test crosses. Test crosses in a pea plant, tallness is a dominant phenotype. If you're given a tall tea plant, how can you find whether it's a homozygous dominant or heterozygous dominant? Because when we say someone is tall, there's a chance it could be homo, which is the same dominant, or hetero, which is one dominant and one recessive. So how do we know? How do we find out? To find out the genotype of an organism showing the dominant characteristic, we carry out a test cross. A test cross always uses a homozygous recessive individual. If the plant is homozygous dominant, the offspring will be tall. If the plant is heterozygous, the offspring will be ratio 1 tall, 1 dwarf, or a dwarf offspring will appear, as you can see shown in the Punnett square. If the dominant recessive, the homozygous dominant recessive plants were to combine with a dwarf, all the child will be tall. All the kids produced will be tall. Whereas if the tallness is a heterozygous tall plant, there will be a chance that a dwarf child will appear. So now here are some questions that you could pause this video and understand, and we'll quickly here talk about condominance. In condominance, both alleles are expressed in heterozygous. What is condominance? So in condominance, both alleles are expressed in heterozygous. Its phenotype has intermediate between that of the purebred parents. Example, when a red-haired bull mates with a white-haired bull, their offspring has both red hairs and white hairs, called roan. This is an example of a condominance. That means both of the parents are heterozygous. It means both of them have different alleles. So like in this case, in the plant case, the second parent, it was homo because they both have the recessive. So let's imagine that both of them were heterozygous. This could mean that there will be, if they were to produce 4 childs, half of them would be white haired while half of them would be red haired, or their offspring have both red hairs and white hairs, called roan. This entire thing is an example of a condominance. Let's move on to hear some more questions using distinguishable symbols to explain the above crosses. You can see the red-haired bull and the white-haired cow. I want you to explain this using crosses. Well, this is a work example. Explain it using crosses. Now here's the actual question. Let's practice. A purebred plant with a blue flowers were crossed with a pure breeding plant with white flowers. All the F1 offspring had blue flowers. When the F1 plants were self-fertilized or bred among themselves, the F2 plants produced were in ratio of 3 blue flowered plants and 1 flower plant. Now using this suitable symbol explain the process above. Now this is very similar with the tall and dwarf plants. It's quite similar. All you have to do is write down the the crosses shown in the question. Now this is easy. You can pause this video and try to solve this in your notebook. And we will move on to the next section. 18.6, Inheritance and ABO blood groups. Now this will be quick here, as we are just going to explain the inheritance of ABO blood groups, phenotypes being AB, and O blood groups. So far, we've assumed that there are only 2 possible alleles for a given trait. In a population, there be more than 2 alleles for a given trait. Multiple alleles is the term used for a gene that exists in more than 2 alleles. Example, the genotype of the allele will be multiple. Here is for blood groups. The occurrence of blood groups in humans is an example of multiple alleles. There are 4 blood groups in human population, namely A, B, and O blood groups. The alleles for A group, B group, and O group are designed with an I and an A at the top of it, respectively. IA and IB are dominant over IO, but IA and IB exhibit co-dominance. Therefore, individuals with alleles IA and IB will have the AB blood group. The blood groups and their respective genotypes are shown in the table right here. So you could see that blood group blood group A and blood group B are both dominant, while blood group O is recessive. So there's a chance that if you combine blood group A and blood group B, you get AB, which contains both the allele of group A and group B. So let's move on. Here is a work example and a question. Now, the work example is solved in your textbook, your biology test, while the best practice is a question you could solve. So here's the question, and please pause this video and try to solve as we move on to the next section, which is sex determination. So when people mate and they give birth to a person, how will you know the gender of the child? Well, this is also through a process. This section will learn about the diploid cell and the chromosomes and so on. So let's move on. In the human body, there are one pair of sex chromosomes. In males, the pair of sex chromosomes consists of one X chromosome and one shorter Y chromosome. Females have a pair of X chromosomes in each normal body. The remainder of 22 pairs of chromosomes are non-sex chromosomes called autosomes. Now, in the human body, there are one pair of sex chromosomes and 22 pair of non-sex chromosomes. This means that each body cell has one pair of each type of chromosome. Hence, the human body are diploids with 23 pairs of chromosomes. Remember that during meiosis, the chromosomes form each pair separate. Hence, each gamete produced can only have one chromosome from each pair. In the male, the X and Y chromosomes will separate during meiosis. The sperm produced contains either a Y or an X chromosome. In the female, the two chromosomes will separate during meiosis. The egg will contain one chromosome. Remember when we said the sperm and the egg are haploid cells. So that means each of them will only contain one single chromosome, while in the male chromosomes, during the meiosis, the two chromosomes, each pair separates. So random fertilization will enable a couple to have equal chances of producing a boy or a girl. Now, shown in this diagram, the sex determination in humans, you can see that a male can produce either X or Y, while a female only has XX. So, if the offspring, the chances of the child being an offspring are determined through cross table. The female chromosomes are homozygous, while the male is heterozygous, and one has X, one has Y. Now, if both were XX, there's a 50% equal chance that they could give female, and if both were XY, there's an equal chance that it could be a male. So the offspring will have an equal chance of being a male or a female. So what is sex linkage? The X chromosome is longer than the Y chromosome. Hence, the X chromosome contains more genes than the Y chromosome. Characteristics controlled by the genes are found only in X chromosome called sex-linked characteristics. An example of a sex-linked characteristic is color blindness in humans. This explains why there are more males with color blindness with females. And how is color blindness inherited? How is color blindness inherited? For the gene for color blindness has two alleles. The allele for normal eyesight is dominant to the allele of color blindness, but since the male only has one X in his cells, chromosome in his cells, he only needs to have one recessive allele to be colorblind. The female needs to have two recessive alleles to be colorblind. That's why the chances of a male having color blindness is higher than a female. Now, using the symbols to represent the alleles, X, Y, N is normal, while X and Y is normal, while X and Y is colorblind. While in female, XN is normal, XN is also normal, while XN is also normal. Let XN be the X chromosomes with the dominant allele for normal sites, while XN be the recessive allele for N for color blindness, and let Y be the chromosome without the gene. So now we have watched example. A man and his wife both have normal eyesight. They have a son who is colorblind. Using the suitable symbols, explain how the son has inherited the defect and is the chance of their next son will be colorblind. So here are some questions. Here are some practice. The figure shows the chromosomes of a human. Answer these questions, and we have arrived at the end of the chapter. They've asked us what we learned. We've learned about inheritance, DNA, chromosomes, meiosis and meiosis, which is in cell division. We learned about genes, alleles, photosynthesis, the photosynthesis in the nucleus and the one in cytoplasm, the making of mRNA, genotype, phenotype, dominant allele, recessive allele, and condominant. So this is the end of this video. I hope you understand this chapter. We have arrived at the end. Please answer all the questions that we've seen in this PowerPoint. Thank you so much for watching, and we'll see you on